Alright, hello, I'm Melissa Matthews. This is my concept map on plasma membrane. We're going to go ahead and zoom in, talking about the plasma membrane. It's composed of the phospholipid bilayer, as we all know. It uses the electrochemical gradient, and it allows certain substances to come in by being selective permeability, which requires either passive transport or active transport. We're going to talk more about passive than active. The main thing we need to know is active transport uses ATP. Then we have passive transport. Some examples are simple diffusion. It uses cell membrane channels or channel-mediated passive transport, which still is passive, but it uses channels versus just passing straight through the membrane. Going back up to the electrochemical gradient, also described as the concentration gradient, which can explain the movement of water or osmosis. These different concentrations are identified as these following solutions: isotonic solution, hypotonic solution, or hypertonic solution, which just expresses different concentrations with the inside and the outside of a cell, pretty much. Okay, going back up to the plasma membrane, which is made of the phospholipid bilayer, which has hydrophobic nonpolar tails or hydrophilic polar heads. Okay, those hydrophobic nonpolar tails can be made of saturated or unsaturated fats, which would just mean a bent or unbent tail. Plasma membranes can also be described as vesicles, which are just small little circular membranes. Okay, back down to the phospholipid bilayer. They have attached or unattached membrane proteins. Permanently attached are those integrated membrane proteins. Not all IMPSs are transport membranes, but all transport membrane proteins are IMPs, okay? So, going back up to the phospholipid bilayer, which can be described, it's been described in various models, including the sandwich model by Davison and Dinelli, or the fluid monosac model by Singer and Nielsen, which both can explain the phospholipid monolayer, which is that one single layer. Going back up, other molecules that have the hydrophobic and hydrophilic ends are ambiopathic, detergent, or sterols or steroids, and other protein examples there are glycoprotein or the protoglycan. So the transport membrane proteins, which are all IMPs, they're all integrated, include the following. You have the receptor membrane proteins, which help with the surface. There are also surface membranes, which assist in the cell recognition, which glycolipids also help. Then we have the anchor membrane proteins, lipid link proteins, and enzyme membrane proteins. So those are all include different types of transport membranes. You have the temporary connected with the IMP membranes, and those are the peripheral membrane proteins. Those don't fully connect, but they partially connect. Your phospholipid bilayer, those membrane proteins, also have ion channels, which are pore forming, and they allow certain types of ions going through, and that's with the gated ion channels. All right, well, that's kind of explains my concept map there pretty broadly. Pretty much just explaining the plasma membrane and the surface of that and how it all works. Thank you.